Welcome to Carbohydrates Part 5. In this tutorial, we'll start to look at the breakdown of sugars. So let's get started. Because we're talking about the breakdown, we recognize that's catabolism, right? We always think, when we hear that word, we think breakdown, right? We can think exothermic, right? Which lets us know that this is going to be energy released, right? So we break down our food to get energy. Okay, so the first stage is hydrolysis. And that's basically like the opposite of what we've been practicing in the earlier tutorials. We were looking at how we take simple sugars and hook them together through glycosidic bonds to make bigger and bigger sugars. Well, now we're going to go the other way. We're going to break them apart. Once we have them into glucose, then glycolysis happens. So let's look a little bit more closely at this conceptually before we dig into the chemistry. All right? So the first stage of carbohydrate catabolism is digestion. We have to break the sugars into smaller molecules for energy production. With carbohydrates, it's very interesting. When we've been nagged to chew our food, there's a really good reason for that. The digestion of carbohydrates begins in our mouth, not only with the physical grinding and softening, but our saliva has alpha amylase in it, an enzyme, right? So when we see ACE, we recognize that as an enzyme. And the alpha, that makes sense, right? So it's going to cleave those alpha glycosidic bonds, right? But the interesting thing is, as soon as we swallow that mouthful of food and it goes into our stomach, the stomach acid denatures the amylase. So inside our stomachs, carbohydrates, nothing really happens there. We have to wait until the carbohydrates are moved on to our small intestine. And then additional resources of alpha amylase and other enzymes are secreted by the, amylase from the pancreas, and then the small intestine brings in maltase and sucrase and lactase to continue the hydrolysis of the disaccharides. So hopefully this will, it always reminds me to chew carefully. So in this tutorial, we will look at the hydrolysis reactions, and then we will look more closely at glycolysis. So basically, the whole point of hydrolysis is to get to glucose. Taking those polysaccharides and breaking them down into glucose. Once we have glucose, then we can use the glycolysis pathway to produce pyruvate. And then we'll see that pyruvate can be converted into acetyl-CoA, and it enters the citric acid cycle to produce CO2 and reduce coenzymes. So this is a pathway that we will be looking at more closely when we get to our bioenergetics chapter. So for now, we'll focus on the part unique to carbohydrates, the hydrolysis of carbohydrates and glycolysis. The stage 3 is shared by proteins, carbohydrates, and lipids. So we'll wait till we've worked through all of the biomolecules, and then we'll look at the bioenergetics. So where is all this happening? All right, well, the hydrolysis we've already talked about. It's happening through our digestive system, but at the cellular level, glycolysis, right? Glycolysis will happen in the cytoplasm, specifically in the cytosol of the cell. And so glycolysis is really important because it's happening just in the fluid portion. So we do have cells that don't have mitochondria. And we will learn that mitochondria, that is where stage 3 takes place. So for the cells in our body that don't have mitochondria, glycolysis is a big deal because that is the primary pathway for energy production inside that cell. All right, so we've looked at catabolism in terms of where it's happening in the cellular structure, and to be more specific, make sure everyone knows the mitochondria is right there, so that's stage 3. All righty, so step 1, where is this happening in the cell? Step 2, let's think about this in terms of the biochemical pathways, right? So the carbohydrates, we can think of those as the polysaccharides, right? And so stage 1 then here will be the hydrolysis, right? Producing the monosaccharides, and in particular glucose. And we're going to be talking a lot about glucose, so it's going to be very handy just to symbolize it with a G. All right, so there's stage 1. And so we'll talk about hydrolysis in this tutorial, and then we'll look at glycolysis, which is stage 2. And then the end result of glycolysis, right, is the pyruvate. So that's, right. And then what we see, right, is so there's carbohydrates. And then notice lipids can produce pyruvate, or they can go straight to acetyl-CoA. And proteins can also be broken down to produce pyruvate or acetyl-CoA. So then that will lead us to the bioenergetics chapter, where we'll focus on stage 3. So stay tuned for bioenergetics. And for stage 2, just to really reinforce, right, glycolysis is stage 2, and we will look at it, we'll look at the entire pathway because there's 10 steps, right? So the 10 steps, sometimes they're really overwhelming. So at a very fundamental level, just keep in mind glycolysis is D-glucose to pyruvate and energy. All right. Okay, so now let's take some time to look at stage 1, hydrolysis. So remembering, right, so our polysaccharides, and keep in mind, right, that if we're looking at starch and things, we're looking at those the alpha linkages. Those are the ones that we can digest. And so here's amylose, a polymer, amylase, the enzyme, right, with and this is a hydrolysis reaction. So there's our enzyme, right? Here's our polysaccharide, and then of course our water, because this is hydrolysis. And so we start to break apart the polysaccharides into monosaccharides, disaccharides, oligosaccharides, dextrins. So let's practice this. So in the previous tutorials, we linked them together. Now let's practice taking them apart. So whenever we're working with sugars, it's always a good idea to find the anomeric carbons. They help us keep our bearings. So we look for the carbons bonded to 2 oxygens. And then when we're focusing on the left, right, think of the anomeric carbon was also the carbonyl carbon, so we can treat it the same way. We're going to break right here, right? So here's our glycosidic bond coming off of the anomeric carbon, and we're going to break it right there. Okay, so now it's just a matter of not losing anybody. All right, so we rewrite the sugar on the left, making sure we include all of its carbons. And then there's our anomeric carbon. And since it was pointing down, we'll go ahead and point down. And then we'll just make sure that we don't lose any atoms, and we maintain the structure of the R group. Now here, we bring in the molecule on the right, and we'll just keep the configuration there. There's our other anomeric carbon. Here's our flagpole, and now just, right, so carbon 2 is down, up, and down. Alrighty. Okay, so we can look here and we see, so this is, hopefully by now you recognize this as maltose. And we see that maltose and then these monosaccharides, by now you should recognize them, that they are both D-glucose. So maltose is a disaccharide of 2 glucose molecules. And recognizing that when the water came in, think about the water, maybe we should write that out, right? So water, it's helpful to think of water as HOH. So here was the OH group of the water, and there was the hydrogen of the water. So the water came in and split apart the maltose, and just like with this, so the anomeric carbon on the left, just take all your knowledge of carbonyl carbons and put the OH with the anomeric carbon. Alrighty, now we're on for a challenge. Let's do the hydrolysis of sucrose. So test your knowledge. Can you draw sucrose right now? If not, you want to be able to by the exam. So let's go ahead and draw it out. Alrighty, so remembering that it's a alpha, right? So if we think about sucrose, right, it's a alpha 1, 2. So there is our, right, so alpha ants are on the ground. We'll go ahead and make sure we keep all our carbons. No sense losing points for lost carbons. And then we go to the second carbon, and by now you should remember that's fructose. So fructose is kind of funny because it has the 2 flagpoles. Alrighty, so with sucrose, right, we have a glucose molecule. So down, up, down, and then we have our fructose, up, down. All right, so we're going to bring water in, and we would have our enzyme would be sucrase, right? The ACE ending tells us it's an enzyme, right? So we think about water, remembering that the hydroxyl group will go to the first anomeric carbon, and then the second H will come over here to bond with the alcohol, right? So when we look at the bottom carbohydrate, it's just a very fancy alcohol. So there's the R group. Okay, so now so when we split this apart, right, just like carbonyls, we just go immediately to the right of the anomeric carbon, and now we rewrite the molecules. Okay, so here, right, that this anomeric carbon, this is where the water comes in, and the hydroxyl portion of the water goes to the anomeric carbon. For the other monomer, get our carbon skeleton in, right? So we're right here, right? So the oxygen was an original part of the sugar, and it's the hydrogen from the water that goes in. And now we'll just make sure that we haven't forgotten any of the details. There we go. Okay, so we've just shown the hydrolysis of a sucrose molecule. So, and we could name the products. By now we should be able to recognize going down. So this would be alpha D glucose, and then here the OH group is pointing up, so this would be beta D fructose. Alrighty, so that's stage 1. So stage 1 is the hydrolysis of glycosidic bonds, and so we practiced that synthesizing those and reversing them. Now we'll go on to stage 2 of carbohydrate catabolism, and that will be glycolysis. So remember, right, that we're going to look at all 10 steps now, so it can be pretty overwhelming. But in a nutshell, it's glucose to pyruvate, right, plus energy, all right? So that's the big picture. Now let's look at the details. So, here we go. Here is our buddy glucose. Notice that initially we talk about how we get energy out, but initially we're actually having to use some ATP. So in the early stages of glycolysis, we talk about that being an energy investment phase, because we're using ATP, so we're taking some of our energetic molecules and consuming them. What is happening during this first step? Well, if we look at glucose and we look at the next molecule in the pathway, we see glucose 6-phosphate, right? So glucose 6-phosphate. So what happened, right? We look at carbon 6 and we see what happened. That the phosphate group from the ATP was transferred to glucose. So we had a phosphate transfer. Now this step is needed because glucose has the ability to slide in and out of the cells through the cell membrane. So by putting on a phosphate group, we add charge. In our next chapter, we'll learn that cell membranes are very nonpolar. So once we put that phosphate group on the glucose molecule, it traps the glucose in the cell, right? So now it's trapped in the cell. And this is good for the cell, because if the molecules are just going in and out at will, it's hard for the cell to, keep an inventory. So, what type of enzyme would this be? The phosphate group started here and ended here. So this first reaction is a transferase reaction. So we would need a transferase enzyme. Now let's look at step 2. What's changing there? Well, here we have glucose 6-phosphate, and now we have fructose 6-phosphate. So we know the relationship between glucose and fructose. We saw that earlier. Those are structural isomers. So basically what's happened in step 2 is we have done an isomerization. So we would need an isomerase enzyme to convert glucose 6-phosphate into fructose 6-phosphate. Now we bring in a little bit more energy because we are using ATP molecules. And so what happens here? Notice we have an alcohol to a second phosphate group. We would describe this as fructose 1,6 bisphosphate. All right? So what did we do? We transferred, right? We did another phosphate transfer. So that's going to require a transferase enzyme. Alrighty. Now, in step 4, we are going to take this very large fructose 1,6 bisphosphate molecule, and we're going to split it apart. Notice what happens here. It gets split into a glyceraldehyde and a dihydroxyacetone compound. Alrighty. Notice that here we have pretty much all, and if we look at the carbon skeleton and everything, we see a lot of alcohol groups. Notice that we get the formation of carbonyls. So in this step, we split a large molecule into smaller molecules and we created double bonds. So that's our clue that we would need a lyase enzyme. We didn't need any ATP here, and water's not involved. There's no oxidation or reduction. It's almost by default you can recognize that we would need the lyase enzyme here. And the carbonyl bond formation is the clue. Alrighty. Now, it's very interesting, but the two products of the splitting of the fructose molecule, if you count up the atoms here, you will discover that these are structural isomers. And so step 5 is that the glyceraldehyde 3-phosphate is able to continue on the glycolysis pathway. So step 5, we need an isomerase enzyme to convert the dihydroxyacetone phosphate into the glyceraldehyde 3-phosphate so that glycolysis can continue. The other consequence here is now all of a sudden you'll start to notice all these 2's in front of everything, right? Because we took one big molecule, we split it in half, and then isomerized. So now we have 2 glyceraldehydes. So now everything is doubled. So we just show it once, but it's important to recognize that the process is happening to 2 separate molecules. So what we can note then is that for every glucose, we're going to get 2 pyruvate molecules. Alrighty, so let's go on with our glycolysis pathway. Okay, so in step 6, what has changed? Glyceraldehyde here. What do we have? Let's see here. Oh, okay. So here we have an aldehyde, and now notice here we have some sort of phosphate ester carbonyl. And we see we have coenzymes. So our coenzymes are reducing and our carbon is oxidizing. So we would need an oxidoreductase enzyme. And then if we continue looking at the changes, now we can see that we're producing ATP, right? So this is where we get the energy payoff. Now we put in 1, but we're going to get back 2 here because what do we see? We have, right, the phosphate group is getting transferred to the ADP. So this would be another transferase enzyme. And then if we look at step 8, what's happening there? We look for changes in the molecule. And what do we see here? This group's the same. Basically, we swapped, right? The alcohol and the phosphate group swapped from being inside and on the terminal edge. So another isomerase enzyme is needed. And then we get, what happens here is we get the formation. If we look right here, we have the alcohol in a single bond, and then notice that we formed a double bond and we lost water, right? Dehydration, right? With carbon-carbon double bond formation. So that is a clue that we would need a lyase enzyme. And then for our last step, we produce more ATP, and if we look at the change from the 2-phosphoenol pyruvate, we see what happened, right? We lost, right? We lost our phosphate group. It got transferred, right? So transferred to ADP, right, to produce ATP. So we get more, right, so we get more energy back. So one last transferase. Alrighty. Okay, so we've gone through the glycolysis pathway in a lot of detail, and I've highlighted all the important points that I want you to know for quizzes and tests. And now we'll just do a little wrap up to see what happens to pyruvate, right? So stage 2 is done, but we have this little transition, right? Because the pyruvate doesn't enter the citric acid cycle directly. So let's look at the fate of pyruvate. There are 2 possibilities. in anaerobic, of course, there's no oxygen. Anaerobic, there is. So, under aerobic conditions, the pyruvate will form acetyl-CoA. So we can look here and we can see that this carboxylate group, right, is oxidized to CO2. So there's our oxidation. And in the process, we get acetyl-CoA, which can enter the citric acid cycle, right? So I've described it to you, and then here it is paraphrased. It talks about, right, where it's happening in the cell and that sort of thing, right? So in the mitochondrial matrix, the pyruvate is oxidized to CO2, and the acetyl group goes to it that form acetyl-CoA, and then it has to diffuse into the mitochondria from the cytosol, and then on the mitochondrial matrix, that's where the citric acid cycle will occur, and that's what happens, right? What happens to the acetyl-CoA? It enters the citric acid cycle. Okay, and so this is where we'll get to, right? So this will all be stage 3, and we'll talk about that in our bioenergetics chapter. We'll go into that in a lot more detail. It's the anaerobic part that we're really only going to talk about on this page, and then it's done. So it's really important to understand the fate of pyruvate. So under anaerobic conditions, right, we think about that as a reducing. So if we look over here, we have a ketone, right? It reduces to an alcohol. So there's our reduction reaction, and that forms lactate. So that's what happens, right? If we're exercising and we can't get enough oxygen to our muscles, right, the glycolysis produces the pyruvate to give us energy, and if there's not enough energy in our oxygen in our body, we end up producing lactic acid in our cells, and that leads to the soreness we feel after heavy physical exertion. All right? So let's look a little more closely, right? So the lack of oxygen will slow down the electron transport chain, and that's part of stage 3 that we'll talk about soon. So without the electron transport chain, we get a buildup of NADH. And we have decreasing NAD+, and we need that for glycolysis. So without NAD+, glycolysis can't continue. So what does the body do? It starts reducing the pyruvate to produce additional NAD+, right? So here's the normal glycolysis pathway. So we can see how they're related. And so then what happens here is, if we have to, we'll reduce the pyruvate, which happens here, to get the NAD+, which then can be used to fuel additional glycolysis. All right. It's interesting to note that tissues with low oxygen content, such as skeletal muscles, they pretty much rely on the anaerobic, I got a typo there, they rely on anaerobic production of ATP by glycolysis. And then as you go into your micro classes, you'll learn about how bacteria can convert pyruvate to lactate under these anaerobic conditions, and that's the chemistry behind kimchi, sauerkraut, and yogurt, with the bacteria that have the enzymes for those conversions. And then those of us that enjoy alcoholic beverages, right, the yeast can convert the pyruvate to ethanol and CO2 under anaerobic conditions. So that concludes this tutorial on the first 2 stages of carbohydrate catabolism. And stay tuned for our bioenergetics chapter for stage 3. Now would be a good time to work a few homework problems to reinforce your understanding with these important concepts.